Okay, good morning, everybody, and welcome to the session of this theoretical computer science section of the ICBS. So it's a great pleasure this morning to welcome Professor Guoan Zheng, who heads up the lab of Smart Imaging Laboratory at the University of Connecticut. And among other things, he is an expert in advanced biomedical imaging. And this morning, he's going to tell us about Ptychography for lensless computation imaging in microscopy and endoscopy. So let's welcome him.
Thank you for the introduction. So again, my name is that too loud? My name is Guoan Zheng, and in today's talk, I want to discuss a new technology called ptychography. And the idea here is we try to perform How can I deal with this? Okay. Are we good? Okay, so the idea here is we try to perform imaging, but we try not to use any optical lens. So that is the basic idea. So I want to talk about the application in microscopy and endoscopy. So try to give you the background. So why we try to do this imaging without using any lens. So I want to show you a very famous image. So my question to the student here is, do you recognize this image? So this image is related to one of the greatest discovery in biology in the past 100 years. Anyone know about this image? Yes, great, good. But you are not a student. So this is the image by the lady, Rosalind Franklin. So this is the very famous photo 51. So the idea is they try to use the X-ray to illuminate the DNA crystal. And they're going to capture the image without using any lens. So you may ask me why they don't use any lens. The reason is that for X-ray, there's no lens. So that's the reason. There's no lens you can use to perform this imaging. So they take this picture, right? And based on this picture, they try to figure out what is the object looks like. And unfortunately, this lady, she cannot figure this out. So this will lead to the Nobel Prize in 1962. So by James Watson. So he figured out this double helix structure based on this lensless image they capture. So you can see here there's a mismatch here. So you capture the diffraction pattern, but you try to figure out this structure of the object, right? Which is not easy by that time. That's why he can get the Nobel Prize. So in this talk, I want to convince you that by using this ptychography technology, we don't need to infer the structure. We don't need to try to figure out the structure. We can directly image the specimen. So there's no way, you don't need to think about what will be the best structure to generate this image. You can directly recover this double helix image based on this diffraction pattern. So that is the motivation here. So the outline of my talk, so first I want to talk about the concept, the imaging model, and the typical experimental setup for this ptychography technology. And I want to talk about different implementations. Like for the student, like you can even hack a Blu-ray drive. So you can buy a low-cost Blu-ray drive. You try to repurpose the Blu-ray drive to perform this ptychographic imaging in your home. We also want to talk about the label-free imaging. Without using any stain or labels, we try to get a good contrast of the sample. We try to combine this with a full cytometry. So we want to talk about this optofluidic ptychography on a chip. Also want to talk about some extension. I want to talk about this temporal correlation to perform high spatial temporal to perform with resolution to perform imaging with a high spatial temporal resolution. Try to perform this depth multiplex imaging. Try to replace the confocal microscopy. We try to get the 3D information of the sample. And lastly, I want to extend this to endoscope. So not only for microscope, we try to apply the same concept for endoscope imaging. So let's get started. So the first point I want to talk about is this the concept, the imaging model, and the experimental setup. So I guess the first question you might have is like, what is this word? So this word comes from Germany. So P here is silent. You are not going to pronounce the P here. So just like psychology, right? So the pronunciation is like ptychography. There's no P here, ptychography. And the meaning here is this tycho means the convolution. So the idea is that by using this technology, you try to use convolution to recover the original object. And this is the original paper. It's about 50 years ago. This is the first concept, but the modern development is substantially different from the original concept. If you look at different wavelengths, so this is the electron ptychography. We can go down to like 1 picometer, like try to pushing the limit. We also have the X-ray ptychography, can push the limit to nanometer scale. And EUV, extreme ultraviolet, is very important for wafer inspection. And you can push the limit to like 20 nanometer. And also we have the optical ptychography. So in this case, we are going to use the optical wavelengths to perform imaging, and we have many benefits of doing that. And you can see that this technology can really cover like a large amount of scales from like picometer to 1 meter. So this is just the development we try to summarize in the recent review paper. So this is the original concept, and the technology took off in 2004. And from there, there's different branches. Like this is the X-ray, this one is the electron ptychography, this one is the EUV, and this is the optical ptychography. And if you read the recent paper on ptychography, you find a lot of Nature Science paper published. Like, for example, people try to push the resolution limit with this electron ptychography. And the idea is, again, without using any lens, actually you can get better resolution. So this is the conventional setup. So this is the concept of this ptychography. So what you have here is very simple. You have a beam, you have a light. This light can be like photon, X-ray, EUV, optical light. It can also be electron. Like you can generate the electron by using the electron gun. So what you have is you have the object. This is the object. And you try to confine the beam into a small region. So you have a pinhole here. So you illuminate this object with this pinhole. And then you put the detector at the far field to capture the diffraction pattern. And in this case, you need to translate this object to different position. And then you can capture a sequence of this diffraction pattern without using any lens. So you can see here, there's no optical lens involved in the setup. And based on the pattern you capture, you try to recover the high resolution image of the original object. So if you look at the imaging model, so what you have is the old object, right? You have the object in the real space. You have X, Y in the real space. And then you have this confined beam. So that's we call this a pop. So you multiply the object with the pop beam. And then you do the Fourier transform. The idea is that the far field propagation here is equivalent to performing a Fourier transform. So you do a Fourier transform, you take the absolute value because you can only detect the intensity pattern. So you do the square, so that will be the intensity pattern you capture. So you can see here, there's several points here. You need to mechanically scan the object to different positions. The image is captured in the Fourier space, which is it's not the real space, it is a Fourier space. Means that you need to propagate the light for a long distance. And then you need to use this confined aperture to generate this pop beam. There are several limitations here. The first one is the limited resolution and throughput. So for this conventional setup, the resolution is limited by this collection angle, and the throughput is really limited by like you need to scan the sample to different precisions, which is like very slow compared to the optical microscope. And also you need to recover both the object and the probe beam. So the O is unknown, right? You try to recover the O. Also, you need to recover this probe beam at the same time. And also you need to capture the high dynamic range the diffraction pattern, like most of the energy concentrated at the center. So that's another problem here. So my talk here is try to address all of these limitations. And by doing that, we can really compete with the conventional optical microscope and even have a better throughput resolution and can also get the quantitative contrast. So that is the motivation. So now I want to introduce our technology, and it's called coded ptychography. So it's very straightforward and simple. I want to show you how to make the device from scratch. So this is my student. So he sacrificed, he made a sacrifice by pooping his finger with the needle. So you can buy this needle from the CVS store from like drugstore. Right? This is for blood sugar testing. So you just get the needle and pop the finger. You get a drop of blood, and then you try to smear this drop of blood on top of the image sensor. So this is the conventional image sensor. What you are going to do is you just put this drop of blood on top of the image sensor. So this is we call this coated image sensor. So we have an image sensor there, and then you have a very thin layer of blood cells on top of the image sensor. We are not trying to image the blood cells. We are using this layer of blood cells as the computational lens. So that is the important trick. There's no other optical lens here, but we use this layer of blood cells as the computational lens. And with that, we can have many different benefits. One immediate benefit is that you can make this layer as large as you want. You just smear the blood to, for example, a letter size gas window. So you have a huge lens compared to the conventional optical lens. So here I want to show you the operations of the prototype device. So what you have here, you have a laser beam. It can be laser, can be EUV light, can be X-ray, and can be electron gunning. So we have the beam coming from the top, and you have the object. And then you have the coded image sensor under this object. So this is, again, the coded image sensor, and we just put the coded image sensor under the sample. So here you can see the operations of the platform. So you can see we're going to scan this coded image sensor to different positions. And then in this case, we try to capture multiple images, multiple diffraction patterns of the object, and then we try to do the reconstruction. Just like the DNA image I showed you at the beginning. So this is the image we capture by using the platform we just developed. Again, this is the diffraction pattern. My question to you is, can you see what is the object here? It's very difficult, right? So this is the recover object. Again, this is the diffraction pattern because we don't have any lens, right? So we just capture a pattern, the light will just diffract to everywhere. And based on this light speckle light image we capture, we can then recover the super high resolution images of the object. And the sample here is the resolution target. So this is just another example. So this is a biological sample. So this is the diffraction pattern we capture. So this is the recover phase informations of the object. The phase information is related to the thickness of the object, like the height of the object. And so this is the unmapped phase, and this is the conventional optical microscope. You can compare the lensless approach with the lens-based approach, and you can see that we get a better contrast. We can also get potentially get a higher resolution. I also want to compare our approach with the conventional optical microscope. So this is the conventional optical microscope. So you have the sample. So you have the objective lens. You have the pupil aperture at the back of your objective lens. And then you have the tube lens, and you have the image sensor. So essentially, this conventional optical microscope is doing two Fourier transform. So the first objective lens is going to do the first Fourier transform, and the second tube lens is going to do the second Fourier transform. So if you look at the image model, so you will do the first Fourier transform to get the object spectrum, and then you will multiply this object spectrum with the pupil of the objective lens. This is the pupil. And then you will do the second Fourier transform, and then you take the absolute value, do the square. So that will be the intensity image you capture by using the conventional optical microscope. And the key point here is if you do two Fourier transform of the sample, you get the original object. So that is the trick of the conventional optical microscope. You don't need to do any reconstruction. If you do two Fourier transform in sequence, you are going to get the original object. I mean, in the sense that they are flipped left to right, upside down. Okay, if you do two Fourier transform to the object, you get an image that is like flipped, like upside down, left to right. So let's take a look at what we are doing here. So we have the object. We have this layer of bud cells serving as the computational lens, and then we have the detector at the back. So what we are doing here is we try to replace the two lenses, this tube lens and the objective lens, with the free space propagation. There's no physical lens anymore. So we have like free space. We just have air between the object and the coated surface, and also the air between the coated surface and the detector. In this case, the image model, so we are going to propagate. We are not going to do the Fourier transform because we don't have a lens. So we're going to propagate the light for distance d1. So this is the d1, the distance between the object and the coated surface. So we get the object wave W at the coated surface plane. And then we will multiply this object wave with the coated surface. This is the layer of the bud cells. Just do this multiplication here. And then we will propagate this light wave for distance d2. And then we perform this intensity detection. So the trick is that based on the intensity measurement, we are going to recover the W, the object wave at the coated surface plane. And with that object wave, we can back propagate this to any position we want to recover the original object. So that is the basic idea of this technology. The take-home message here is that this layer of bud cells is serving the purpose of the computational lens. The question I have here is that which type of bud is the best for this technology? So for example, we can use my bud, human bud, right? We can use fish, goat, duck, and chicken. So which type of bud cells is the best for this device? Any idea? My student use human buds, right? But do you think like other animal can give you better performance, and why? One of the animals is very different. So can you tell that? And why? Turns out the answer is the gold bud. And the reason is the gold bud is the gold bud cell is the smallest among all animals on the earth. So the size of the gold bud cells is 2 to 3 microns. So this is the recover. So this is the performance, right? This is the diffraction pattern we capture. So this is the recover image by using the gold bud cells. And this is the recover transmission profile of the gold bud cells. And you can see the size of the cells is about 2 to 3 micron, substantially smaller than human bud. And we also test the fish bud we mentioned. So this is the fish bud. It's very difficult to get the fish bud in the United States. You need to go to like stores in New York. Like we drove for like 3 hours, go to New York, and they are going to kill a fish that we can collect the buds. It's very different from China. Like you can just go to any market, you can get the fish bud. So you can see the size of the fish blood is substantially larger, and that's the reason it's not a good computational lens. And the take-home message is that you need to use very small particles to serve as this computational lens, so that you can capture more informations in the Fourier space. Because this, if you use a very small particle, it can deflect the light. So those high-angle light can be captured by the system. Another question is like, can we use any crab to make a good coated surface? So this is, for example, this is the farm on our campus. So my student collect a piece of crab, like he put this on top of the image sensor, and then he make a smear of this piece of sheet on top of the image sensor. And this is the reconstructed intensity and the phase image of that piece of crab. And this is the recovered resolution target. And you can see here, the performance is not good. And the reason is that it's not uniform. Like there's an empty region. And also this piece of crab is too thick. And you can see many phase gaps there. Means that it's not a single layer. So the take-home message is that we need to have a uniform thin layer to perform this modulation. So what we did is like we developed a process to do that. So we have these microparticles on the image sensor. So this is the final device we make. Without using the bus cells, we can just use these nanoparticles. Put these nanoparticles on top of the surface. So this is the prototype we make. So this is the best performance we can get. We can resolve this 300 nanometer line width on the resolution target. And again, this is the diffraction pattern we capture, the raw data we capture. So this is I guess the video to show you the operations. So we can, different from the conventional optical microscope, we can make an array of this device. So we can do the scanning and we can image these pathology slides all at once. So we get about 0.8 numerical aperture without using lens. This is one of the highest resolution for lensless imaging. We get about we can resolve a 300 nanometer line width. We capture like 200 images for each sensor. The acquisition time is about 8 to 15 seconds, and we can image an effective field of view like larger than 240 millimeters. It's about 2 orders of magnitude. 3 orders of magnitude larger than the conventional optical microscope. So another advantage here is we can image the phase information, which is related to the height of the sample. So for example, here, we can recover the phase of a different target, and you can see that they match with the ground truth. So this is the phase information of an optical prism. This is the lens. This is the phase information of the bacterial colonies. Another advantage here is that once we recover the light wave information at the coated surface plane, so this is the recovered light wave at the coated surface plane at that basal layer, we can propagate the light to any position we want. And in this case, we can propagate it to the in focus position to recover the high resolution image of the sample. So this is the demo here. So this is the huge pathology slides, and this is by using our lensless platform. So you can see you have a very large imaging field of view. And the key point here is that the size of the field of view is no longer limited by the optics. There's no optics anymore. So you can image a large section just using a large image sensor. And also, you can get very high resolution compared to the conventional optical microscope. So this is the difference. So what I want to show you here is a video to show you the image we cover. So this is the pathology sections typically used for medical diagnosis. I mean, the gold standard is that the doctor need to take some tissue samples from the patient, and they try to put this on top of the glass and try to image this section to figure out the type of disease of the patient. And you can see here, you can zoom into different parts of the sample, and you can still able to see the very fine details of the of the cells. So the entire image contains like more than 10 gigapixels by using this lensless imaging device. So we can also perform like blood cell differential counting. So this is again 1.8 centimeter imaging area. And you can zoom into this very small, very tiny pot, and you can see those white blood cells. So that is the phase of the white blood cells. So those are the intensity of the white blood cells. You can also compare with the conventional optical microscope, like 40x objective lens. You can see that they look very similar to each other. From there, we can perform like this white blood cell differential counting. We can count like how many types of white blood cells, the ratios of the white blood cells with the blood sample. So another direction we try to push is we try to compete with the conventional interferometer to perform optical metrology to measure the nanometer scale surface of the optical elements. So for example, this is by using our lensless imaging device. So this is using the very expensive cycle interferometer to measure the nanometer scale roughness of these freeform optics elements. And we can compare the results, and they match with each other. Another commercial direction we try to target is try to perform this optical metrology for this medical device. So this is the transdermal microneedle patch. So you can deliver vaccine with this patch without using the needle. You just put this there's a bunch of micro needles there. You just put this on your skin, and it will deliver the vaccine to your body. But in this case, we need to characterize the 3D shape of these micro needles, and it's not easy to do by using the conventional optical microscope. But what we can do here is we can recover the phase images of this microneedle patch, and from there we can unwrap the phase, and then we can get the 3D measurements of these microneedles. So again, this is the phase image we recover, and this is the unmapped phase, and from there we can get the precise shape of this microneedle, which is difficult to do using the conventional approach, or it is impossible to do with the conventional optical microscope. So this is again one of the microneedle. This is the unmapped phase, and this is the conventional optical microscope image. You can measure you can tilt your sample, try to measure this microneedle out at the outest of the patch, but you cannot measure the shape like within the patch. So this is the 3D image recovered by using our lensless imaging device. So with that, I want to talk about different implementations. So one fun project here is we try to hack Blu-ray drive. From there, we can develop this device like based on the Blu-ray drive. So this is the Blu-ray drive we bought from eBay. So it's about $60. So in this Blu-ray drive, actually you have all the components you need. So the first thing you need is you have a light source. You have 2 laser diodes within that Blu-ray drive. So you have a blue laser diode, you have a red laser diode. You can just control try to give the voltage, and you can control the laser diode. Also, you can have this rotational stage, and you have a translational stage. So based on this, the Blu-ray drive, you have everything. So this is the prototype device developed by one of my undergraduate students. So we just repurposed this Blu-ray drive for this high throughput lensless ptychographic imaging. So what you have is you have the blue laser diode to illuminate the sample. And then we are going to rotate this sample, translate the sample to capture multiple diffraction patterns of the object. And then you can just switch to another sample by using this rotational drive. And then repeat the process again. So this is the performance. So this is the recover image by using this Blu-ray drive modified device. We can resolve like 400 nanometer on the resolution target. So this is the raw diffraction pattern we capture. We can also recover the quantitative phase and the 3D measurements of the sample. So this is the test on the bacteria. So you can see this staph bacteria is very small. You can resolve the individual bacterial cells over a very large imaging field of view. One application we try to target is try to do this urine analysis. So this is the urine sample collected by my student. So basically, if you look at the sample, the urine sample of yourself, you can find a lot of different stuff. So this is different types of crystals. So this is more crystals, and there's some other unidentified stuff in your urine sample. So this calcium oxalate. This is particularly important because this is related to the kidney disease or kidney stone. So if you can find a lot of these calcium oxalate crystals, you need to like make an appointment with the doctor to examine your kidneys function. And the idea here is that based on the large field of view, so we can locate those calcium oxalate crystals automatically, like from these gigapixel images. And from there, we can know the numbers of crystals we can find from the sample, which cannot be done by using the conventional optical microscope. Recently, we also try to send the device to the International Space Station. And the idea is that in the space station there's a space limit. There's also the weight limit. You cannot use a large microscope there because, like, if you occupy the space, which is like unacceptable for those applications. So what we did is we try to make a handheld device. So what we are doing here is we have this voice actuator to translate the image sensor, and then we can make this a really handheld device. So this is the operations of the device. So what you can see here is we have the laser diode, and we have this mirror to try to like extend the beam. So we turn on the laser diode, and the beam will go to this mirror and illuminate on this. This is the coated image sensor. And we have the whatever object you want, like from the conventional microscope slice to like cell culture to any object you want. And we just close the lid so that we can illuminate the object. And from there, you are good to go. So you can capture the diffraction pattern, and then you can recover the high resolution image. And we test the device like in the field. So this is the diffraction pattern we capture. My question to you is, do you know what is the object we try to image here? So this is not in the lab. We take the device out because this is a really handheld device. We take the device out and we capture the diffraction pattern of the object. Any idea? And this is the recover intensity image, and this is the recover phase image. So what is the object we are trying to image here? No? No idea? No. It's a snow crystal. So this is maybe a better one. So we have the raw data, we recover the intensity, and try to recover the phase information and get the 3D shape of the crystals. Like when there's a snowstorm, we just take the device out to perform this measurement. We can also put the device into the incubator. So for example, we try to monitor the cell culture over time. So what you can do is you put this compact device within the incubator. You have the cell culture dish pasted on top of the coated sensor. So this is the very large imaging field of view. If you zoom in this very small part, you can see the cell migrations over time. And you can also have the 3D measurement of the cells based on the recover phase images. You can see this nano is called tunneling nanotube for these stem cells. So we can also perform like the wound healing. You can monitor the wound healing progressions like within the incubator. We try to also perform this label-free imaging. So the idea is that we are not going to stain the sample, and we try to get the good contrast from the object. So there were two options. The first one is we try to do the polarization detection. So the idea is that we try to detect the changes of the polarization to figure out the properties of the object without any external staining. So this is the device we make. So the idea is that we put ultra-thin polarizer films on top of that coated surface. And then we can rotate the entire coated sensor to different angle to detect this polarization-related properties of the object. So this is a 4 sets of measurement by rotating this coated sensor to different angle like 0 degree, 45, 90, and 135 degrees. And we can recover these different images at each orientation. And from there, we can recover the by refringing properties of the object, which related to the refraction index under different polarization states. So this without any staining, of course. So this is the optical X orientation, retardance, and this by refringing map. So we can again we try to get a large imaging field of view and high resolution at the same time. One application is we try to do this malaria-infected blood cell screening. So this is the raw image, and this is the recovered by refringing map of the malaria-infected red blood cells. And you can overlay it with the face image, and you can see those bright spots. They are related. They are related to the malaria parasites within the blood cells. We also try to do this cancer screening. So this is the thyroid smear from a clinical patient. So what we can do is we try to recover this by refringing map, which correlated to these tumor regions of this thyroid smear sample. We also correlated this with the fluorescence image, so we can see the correlation between the fluorescence labeling and also this polarization-based label-free reconstruction. Another development here is we try to push the wavelength to a shorter limit. So what we try to do is we try to implement this deep ultraviolet coated ptychography using the deep ultraviolet light. Typically, is less than 280 nanometer in wavelength. And the reason is that at this frequency, at this wavelength regime, the sample, the biological sample, has the intrinsic contrast because the DNA will absorb this deep UV light. So you don't need to do any staining. You should be able to still see the contrast. And it's important for a cancer diagnosis because you need to do this diagnosis when you are doing operations. You want to make the decision very fast. So we don't want to stain the sample. We try to image the sample and get the results as soon as possible. So what we do here is we try to etch the filters on top of the image sensor. And from there, we can get a good sensitivity to the deep UV light. So this is the diffraction pattern of the resolution target. And this is our reconstructed result. We can resolve about 400 nanometers from the resolution target. So this is the biological sample without any staining. So we can see the cell cultures at this deep ultraviolet wavelength. So this is the intrinsic contrast of our reconstruction. Again, the reason is that the cell, the DNA, will absorb this deep UV light. And for conventional microscope, you can see that the sample, they are transparent because the visible light cannot be absorbed by this the DNA structure. And we also compare with the labeled fluorescence image, and you can see the correlations between them. So this is again for the label-free imaging, and we can try to perform nucleic acid mass imaging. So we can image this is related to DNA, and this one is related to the proteins. Without labeling, we can perform this differential imaging. So this is the nucleic acid mass, and this is the protein mass for this tissue section. So with this new DNA and the protein mass differential imaging, we can perform this virtual staining. So we are not I mean, the sample is unstained, but based on the contrast we capture, we can virtually stain the sample just with the conventional H&E colors. So this is the image captured by the conventional optical microscope. You can see that they are transparent and you don't see any contrast from the object. Recently, we also try to combine this with flow cytometer to perform this optofluorid ptychography on a chip. So the idea is very simple. So what you are going to do is you have this microfluidic channel, so you can translate the object on top of the coated surface. We capture the diffraction pattern, and based on the diffraction pattern, we try to recover the high resolution intensity and the phase image of the object. So this is the chip scale device. So this is the microfluidic channel on top of the coated surface. So you can fold these cells or different objects across the channel. We capture the diffraction pattern for reconstruction. So this is the captured diffraction pattern. So we can recover the high resolution intensity and phase image of the object. We also can compare with the conventional optical microscope. So for these are two different species. So this is just another example. So this is the C. elegans. We can based on the diffraction pattern, we can recover the intensity and the phase information. We can also compare this with the conventional optical microscope. With that, I also want to talk about some extensions of this technology. The first one is we try to improve the temporal resolution. So far, what I have talked about is just a fixed object. We try to, in this case, we try to image the object that is changed over time. So the idea is that in the reconstruction process, we need to consider the temporal correlations of the object. And by doing that, we can substantially increase the temporal resolution. So what you see here is that by considering this temporal correlation, we can image the real-time development of this bacterial culture. So this is a bacterial colonies. You can see the phase of this colony changes over time. It becomes larger and larger, means that the bacteria is growing. So this is the reference time. This is the 30-second growth, 60-second, and 90-second. And again, we have a huge field of view. We can have a centimeter field of view, and you can see the bacterial colonies and different regions. And we can also monitor the growth from the single cell to microcolonies. You can track the height map of this growth. You can see this multi-layer structure of these colonies. Again, you can do this over a large field of view, and you can track different time point of this dry mass of the colonies. One application here is we try to perform this antibiotic drug testing. So the idea is that if a patient gets some infection, you need to determine which drug is most effective to the infection of the patient. What we are going to do is we can monitor the growth of the bacterial colonies over time, and from there we can determine these so-called MIC minimum inhibition values. That is the drug concentration we are going to admit to the patient, giving to the patient. Certainly, we can also do this cell culture imaging. So we can, this is the raw diffraction pattern of the cell culture. And we can also monitor this cell culture growth over time. We can also monitor the melting of the snow crystals. So we can see the changes of the phase over time. So I hope I convinced you that by considering this temporal correlation, we can also perform this video weight real-time imaging of different dynamic objects. And recently, we also try to compete with the conventional confocal microscope. So what we are going to do here is, this is the conventional microscope platform. So for different objects, you need to put this object at different locations to do imaging. That is what people can do with the conventional optical microscope. So what we are doing here is we put different objects together. So we just stack different multiple layers on top of our coated surface, and we change this illumination angle. And from there, we try to do this tomographic reconstruction to recover the object at different layers. So this is what we try to change the illumination angle with this car mirror. And so this is the device. So we have the illumination beam, and then we change the illumination angle by apply a voltage to this motor so that we can tilt this mirror, and we can capture the diffraction pattern. And from the data we capture, we try to recover the multi-layer object. So in this case, we have 4 different layers. Now we have the coated layer, and we have 3 different cell cultures stacked together along the Z direction. And for each layer, we have a very large imaging field of view. And we can also monitor the cell growth over time. So this is another demo. So for 5 brain tumor sample, we just put these 5 sample together along the Z direction, and we can image all of them at the same time. So this is for one section, you have a very large field of view, and you can also compare with the conventional optical microscope. So the last part of my talk is we try to extend this for endoscope application. So I'm going to skip this part. It's about we can also implement these concepts with the conventional optical microscope by putting this coated sensor, by using this coated sensor, by replace the conventional image sensor with the coated sensor. I will just skip this part. And the last part I want to talk about is try to extend the same concept for endoscope imaging. So I want to ask you, what is this image I'm showing you here? Yes, that's the black hole. So this is at the center of the Milky Way galaxy. So this is the black hole image. And turns out that the concept of our platform is similar to what they are doing for this black hole imaging. So for this black hole imaging, the trick is that for each of these telescopes, the resolution is very low. So what they can do is they can use a bunch of these very small telescopes, and they're going to rotate. I mean, the Earth is going to rotate to different direction so that you can have different measurements in this Fourier space. So the trick here is that you're using a very small telescope, and you can synthesize a very large virtual telescope that can resolve this black hole image. So why we can also do that with the endoscope platform? So you consider this is the coated sensor. You can translate this coated sensor to different position, and then you can synthesize a much larger apertures to resolve the details of this object. Just similar to what they can do with the black hole. And what we are doing here is instead of using this coated sensor, we try to do endoscope imaging. So we are going to use this lensless fiber bundle tips. So this is a lens fiber. This lensless fiber bundle actually served it as the coated surface. It can modulate the light by using this bunch of the fiber bundle tips there. And you can translate this lensless fiber bundle tip to capture multiple diffraction pattern, and from there you can recover the high resolution image of the object. This is also similar to this so-called synthetic aperture radar. So the idea is that for the antenna of your airplane, the size is very small, the resolution is very low. So what you can do is that you can capture multiple images when the airplane fly across the object. So you try to detect, for example, an object is far away from you. For example, you are going to detect the airplane from the other country, right? So what you can do is you capture multiple images, and then you can synthesize a much larger apertures. This is the original aperture, which is very small. And after you fly across the object, you can synthesize a much larger aperture with much higher resolution. The same concept here. For each fiber bundle tip, the image resolution is very small because this aperture is very small. And by translate this fiber bundle tip to different positions, we can synthesize a much larger virtual apertures with a much higher resolution. So this is the imaging model. We have the object, we have the lensless fiber bundle tip. We translate this fiber bundle tip to different positions, and then we can capture the diffraction pattern without using any lens. So this is the object. The object propagates to the lensless fiber bundle tip, which is the coated surface plane. we perform the modulation by this coated surface, which is the lensless fiber bundle, and we capture the diffraction pattern. So based on the diffraction pattern, we try to recover this wave at the coated surface plane, and then we propagate this back to the object plane. So this is the video to show you the image acquisition process. So I just hand hold this fiber bundle tip. You can capture a bunch of this diffraction pattern. And based on the data we capture, we can then recover the high resolution image of the object. We also need to correct for the changes of this modulation profile of the fiber bundle tips. So this is just some orthogonal mode decomposition to model the time variation of this fiber bundle tip. So this is the result. So we have the raw data. This is the raw measurement. And then we can recover the high resolution image of the object. So this is the resolution target. And this is the bus smear sample. You can see this is the raw data. And this is we can resolve the individual bus cells. This is the phase of the bus cells. We can also do this in the reflection mode. So this is the resolution target wafer. And this is the mouse colon sample. And we can resolve the very small details on these different targets. And recently, we also try to implement this with this distal chip endoscope. So we try to make a very tiny image sensor chip, attach this to this endoscope probe. So from there, we can perform a high resolution image, bypass the resolution limit of the conventional endoscope approach. So one application here is like once we recover the wave, we can propagate this back to different positions, right? So we can perform this 3D imaging. So this is the 3D shape of a cone object. And similarly, we can also, this is the Lego toy. So this is the recover face information, and then we can get this 3D shape. So color here means the high map of this Lego toy. So this one may be a better visualization. This is another surface. So based on the recover phase, we can propagate this back to different positions. And at the end, we can recover the 3D shape of this surface. And you can see this is the all-in-focus image. And the color here is means the height map of the object. So this is the 3D. This is the 3D images of these different types of objects. And also, we can also perform this resolution improvement. So we just compare with the diffraction limited resolution. So this is the diffraction limited resolution, and this is the sympathetic aperture resolution achieved by using this endoscope probe. This is our result, and this is the diffraction limit image, and you can see the improvement regarding the resolution. So with that, I want to conclude my talk. I hope I convinced you that by using this lensless coated ptychography technology, we can get a high resolution and large field of view. The field of view is no longer by the limit of the optical elements, but is limited by the size of your detector. We can get a centimeter scale long working distance. We can propagate the light to any position you want. We can get nanometer scale sensitivity of the 3D shape. We can compete with the conventional interferometer without interferometry measurements. We can perform label-free imaging with this intrinsic polarization or deep ultraviolet contrast. We can also get high temporal resolution to monitor the video wave changes of the different types of cells and crystals. Can also compete with the confocal microscope by performing this depth multiplex imaging. Lastly, we can also extend the concept for endoscope imaging. So I want to end my talk by showing you how to make this device from scratch. So we send this device to the space station. Thank you.
Related to your last part, so the endoscope, I wonder, you were showing the detector that you were putting on the top, but the endoscope, you may have a cylinder, you may have more larger surface area. Possible to use more flexible sensor in a way that to utilize this cylinder as a sort of a detector, and then maybe also couple with some illumination reflex in a way that can possibly using this detection method to enable even larger area and more sensitive detection.
You're absolutely right. Yes. So right now, the image sensor is the square, but the shape here is the circular. So they do have some gap between for the illumination. So the gap is like you can insert those fiber to illuminate the object. But one design is that you try to change this circular shape to the just square shape to match this image sensor. Or just like what you say, you can have some flexible sensor to do this. Yeah, but absolutely yes. The key point here is we try to demo this to bypass the resolution limit. So that is the key point of the work of the paper, the original paper. But in the long run, there's many possibilities to further improve the device. The fundamental point here is we make that a coated surface. So that coated surface can can modulate the light. Just like you have a very large angle light that carry the high frequency information. And by modulated by that coated surface, you convert this back to a small angle light. You can detect it by your device, just like the conventional optical microscope, but in a sense that you don't need to make a huge lens, you don't need to correct for aberrations, you don't need to make a bulky optics elements. You just make a very thin layer of the surface, just like the metal surface or what they call it. But for us, we just need a random surface that is uniform and thin and contains like very small particles to modulate the light. Yeah, yeah.